Yeah, it's unusual. I had no question today, huh?
It's all right, it works out. I'm busy dealing with people who didn't do the pre-lab. So we are recording. If you're watching this video, there's still like 12 minutes before we officially get started, so you can go ahead and fast forward about 12 minutes into the video.
Yeah, Michelle. Michelle. Yeah, Michelle. Girl, I'm about to hang up on you. Bye, I'm in class. Playing with you. I don't know why everybody think I'm joking.
Did you say that again? I didn't quite hear you.
I didn't say anything.
Morning. Morning. Oh, yeah, you did do the videos, right? Oh, good, maybe it's a mistake. Yeah.
Now that people are coming in, do you understand that? I don't know why that document only showed up through our email. And then when I click on it, it does not show in the base. Like, my independent work showed in there and I didn't submit it. So I don't understand what happened to that pre-lab. And it's looking like I never did it, but me and you discussed it before we could do it.
You know, I don't know. Yeah, you just,
And I submitted everything else, but I just wanted to bring that to your attention. I don't know what,
yeah, it's very weird. I don't understand it.
yeah, because you walked me through because I didn't know how to do it at all. So, and when I was like, I know I did this, like, he was like, oh, don't forget to, it was late. And I was like, no, we talked about it.
Well, the actual lab one.
Yeah, no, lab one, I know it was.
Yeah, well, luckily, like you said, I already knew you did it because we talked about it.
Yeah.
So even though it's late, it's only late as in we haven't figured out how to properly upload it yet. So I know you did it on time. We're just dealing with the technology. So yeah, you'll want to delete that PDF that you have in there, or whatever it is, that,
Yeah,
the graph, and then upload the PDF of your entire lab. in this place.
Hopefully I can get it done right today.
Yeah. I don't know if you're on the Facebook group, but I posted an announcement to the students. I guess I should post it to the WVSU online, but kind of apologizing. I've got to be really picky about how the grade things are turning out, because I have about a little over 100 students. That's about 100 labs to grade per week,
Yeah, I know.
about 25 pre-labs per week, 175 discussion posts, initial posts, and 125 replies.
You know, you give us extra credit on these other.
Yeah, so I just don't have time, that's why I'm really picky.
No, I don't think you did. I think you're very funny with you.
No, the video. Right where I highlighted it and said you didn't do this. Anyway, here we go, let's get started on attendance. Bangura. Today, 29th, all right. Bangura's not here yet. Case Bowl is here. Tristan is online. Don't forget to send those words, Tristan, at the end. Covington Griggs? Yeah. Yep. Just talking. He is here. DeVere? Not here. Poet? Didn't even know it. Dow is here. Hughes is here, right? Hughes? No, that's Thompson. Chad?
Thompson?
Yeah, okay. So Hughes is not here. Lebdy is not here. Carla's here. Mankins, not here yet. Mathis is here. Rains is here. Reed is not here. Elise is not here. Thompson is here. Withrow is not here. Yindui, not here. And Elkins is here. It's 8 o'clock. That was good timing. Let's get started. First word for attendance will be lab. Let's start talking about labs a lot to begin with this chapter, today. So, lab 1 is way behind us now, right? So that was from last week. It's past due. It was the first lab, so if you still haven't done it or haven't figured out how to upload it, that's okay. We'll figure it out. I'm not being very picky with the due dates on that. But with the lab itself, once you're done with the lab itself, not the pre-lab, but the lab itself, you want to download it and then upload that document to My Online. I can show you how to do that if you need help. Some of you couldn't come to lab last week because you didn't get the pre-lab done in time. You've already received an email last week, so you have the regular lab to do, so you're going to turn it in the way I just described. But then you also have to do that other extra lab that I sent you, and you're going to share that with me via Google Docs like you would independent work or like you would a pre-lab. So any questions about lab 1? All right, lab 2. Again, I'm still being pretty loosey-goosey, because we're still learning how things work. But lab 2 is now past due. If you haven't done it, you've received an email. I warned you guys last week before we started, like lab, and then lecture, and then in announcements, and then in emails, like, hey, there's videos you have to do for the pre-lab. A lot of people still didn't, not a lot. Some people still don't do it. So you gotta pay close attention. And it's really highlighted right there. It says, hey, do this before you move forward. And then once you get to the pre-lab questions, it says in big bold letters, hey, before you do these pre-lab questions, make sure you go up and do the videos. So you gotta pay close attention. And also, I'll remind you too, spend 5 minutes, maybe online before you need to turn in an assignment. Let me look it over. Like something like that, I would have been like right away, like, oh yeah, make sure you do this before you turn it in, because I can't grade it. Just you get 0 unless you do that, right? Take me up on that. But again, it's early in the semester, so if you can't come to lab today, it's not the end of the world. You're just going to do the lab anyway. I'm going to collect the data for you, or maybe you get your classmates too. So when we do the lab today, I'll collect the data, I'll share it with you. So then you can still do the lab, and you'll turn it in the regular way you would. You would download it and then upload that PDF to My Online. But as you know from the emails, so hopefully I'm just talking to myself here, so you've already read the email. But if you can't come to lab today, then you also have to do the additional lab. And the additional lab is pretty much the same as the regular lab, except the end. The pH part is different, so that's the part you're gonna have to do. So when you do that extra lab, you can ignore everything else other than the bottom PDF part. And again, the regular lab you're gonna upload as a PDF to My Online, and if you have to do the extra lab, you're gonna do that, share that with me via Google Docs. And of course, the rest of you can still do those extra labs if you want for extra credit. So they have to do it. If they're not here, they have to do it to make up for not being here. And the rest of you can do it for extra credit. Also, don't be late. The last week I was a little bit loose on that. This week I won't be. Don't be late. I'm gonna lock the door if I can remember, because this is a team effort. So when you come in late, then you're gonna disrupt everybody, and it'll take longer for them for everybody because of you. So I'm just being fair to everybody else. So any questions about labs in general? All right, I'm still catching up on grading, so please bear with me if I'm a little slow to respond to emails. As usual, if you need me in a hurry, call me or meet me online for, bless you.
Thank you.
For office hours. Is there anything else? Do you guys have any announcements, questions, reminders? All right, then let's get started. Here's where we left off. We were still talking about water. We were talking about how water is a really good solvent. So there's all these life-giving properties of water, and this was the last one that we were talking about. It's how great of a solvent it is. And I told you, oops, I took this picture. We didn't really talk about it much, but I did explain, and this is just one example, right? Your body needs sodium. Your body needs chloride. But it doesn't necessarily need sodium chloride, right? So when you eat salt, table salt, you're not specifically giving it what it needs, because it needs to break that down so it can use the sodium and so it can use the chloride. And that's why water is so important. Like, so for example, if I gave Carly here like a big old block of red and green Legos, and I said, please build me a green Lego house, what's the first thing you would do? A big block, they're all stuck together, the red and green, I give them to you and say, please build me a Lego house. Yeah, she'd have to break them apart first, right? So she needs these red, whatever color I said, she needs those red Legos, but they're mixed in with the other ones, so she has to separate them. That's the same with this. And that's just one example, salt, right? But there's a lot of other things where your body needs to dissolve things, so it needs that water as a solvent. Whew. Any questions about that? Okay. So this is perfect timing. Now we're going to talk about acids, bases, and pH. This will be important because in your lab, this will be the last thing we do, right? So you're going to deal at the beginning of the lab with things like atomic numbers and valence shells and how many reactions can this thing have, and then you're going to learn how to draw a molecule, and you're going to have some practice doing that. And then what we're going to do is look at some acids and bases. So here we go. Does anybody know what an acid is? Now, depending on your source, there's different definitions, but does anybody, how would you guys define acid? Does anybody know? Before I took this class, I would have said it's something that dissolves something, would have been my answer. And that would be a partially correct statement, but that also depends on a lot of things. So if that's your definition, kind of purge that from your brain. There you go. As far as we're concerned in this course, an acid is a compound that releases hydrogen ions, H+ ions. So you should associate H+ with acid. Now again, this is according to our course. We oversimplify things. That's what you need to know for lab. That's what you need to know for the exam. For independent work, look it up, right? Compare what I just told you to what some other source says. According to the textbook, this is the definition of an acid. According to this source, this is the definition of an acid. And then just write a couple things about how things are similar or different, right? Compare the two. It really helps me to understand or realize that you understand these concepts. Also, it'll help you understand these concepts. All right, so.
You said the difference of the compounds you want to explain?
This, the definition. So I'm telling you the definition, according to your textbook, of an acid is a compound that releases hydrogen ions. So for independent work, you could look up the definition of an acid and then say, all right, here's what the textbook says, here's what this source says, here's how they're similar, here's how they're different. So let's make things simple. Now there's going to be a little bit, it's going to get a little bit more complicated. But when you think of a base, hopefully you think of something that's the opposite of an acid. So with that being said, the first simple way to describe a base, if you look at what an acid is, and think about the fact that a base is the opposite. What do you guys think a base might be? When you're looking at this, and then think, all right, what would the opposite of a compound that releases hydrogen ions be? And if you don't overthink it, this should be easy, but it'll be hard not to overthink that like this.
Something that captures hydrogen ion.
Yes, great. So that is one of the ways we look at it. Thank you, very good. But it's something that accepts those hydrogen ions, right? So acids are like, hey, here's some hydrogen ions. Bases are like, oh, I'll take those hydrogen ions. There's also another way to describe it, and another way that you need to understand for the exam and for the lab. You should associate a base with another ion, but it's a different ion. Hydroxide ions, OH minus. Now go ahead and tell you, I will say these words a lot. I will say hydrogen ion, I will say hydroxide ion, but for the exam and for the lab, you could just be familiar with what it looks like. You know what I mean? Like, instead of remembering that word hydroxide ion as a base, you could just remember that OH minus, a base, or H plus is an acid. So yeah, bases remove the hydrogen ions from a solution. That's kind of how they're the opposite of an acid. And the way they do that is because they're releasing these hydroxide ions. So basically those things balance out. Even if you're not a chemist, I mean, just focus on this, right? Acids have a plus. These bases have a minus. We know those things are probably going to be attracted to each other because pluses and minuses go together, right? So that's how that works. Very basically, that's how it works. As far as we're concerned, that's how it works. So obviously, when you add an oxygen and a hydrogen to another hydrogen, you're going to get water. But again, more importantly, you need to associate H plus with acids and OH minus with base. Are there any questions so far? All right. So now let's talk about how do we measure it. Well, actually, before I put it on there, I'm sure you guys know, so don't be afraid to try to answer this. How do we measure how acidic or basic something is? Like if I was to say, how do we measure weight? We would say, hey, you put it on a scale. How do we measure temperature? With a thermometer, right? The Celsius scale, the Fahrenheit scale. So what do we use to measure acids and bases? Good. That's not what I was going with, but you are correct very often. And I think someone just said it. It's the pH scale. So yeah, I didn't ask that question properly. Yeah, so we would probably use a cylinder to physically do that. But yes, the pH scale is the scale that we use to measure, right? So again, using temperature as an example, Celsius, right? 0 is freezing for water. 100 degrees is boiling for water, depending on some other things. So pH scale is the scale that we use to measure. It describes the acidity, which is the measure of concentration of H plus ions in a solution. But you don't necessarily need to know that the definition. You just need to know it's the pH scale is what we use to determine how acidic or basic something is. And then soon on the next slide, soon on this slide, and then also in the next slides, you're going to learn a little bit more about what exactly these numbers mean. But for now, you just need to know that it's the pH scale that we use to measure how basic or acidic something is. So any questions about that? All right. It ranges from 0 to 14. You need to know that for the exam. You'll get very familiar with it in the lab, hopefully. 0 to 14 is the scale. 0 is most acidic, 14 is most basic. Definitely need to know that. There'll be a couple of test questions where you'll need to know that. You'll have a question where you need to figure something out. And to figure that thing out, you need to know that fact, that 0 is more acidic and 14 is more basic. Alkaline is another word we use for basic. And just think about all the different ways I could say that too. I could say instead of 0 is most acidic and 14 is most basic, I could also just as easily say 0 is least basic and 14 is most basic, right? So there's all these different ways I can describe the same situation. So keep that in mind, because in the exam, your brain's going to need to be flexible and think about it in different ways. Here's another thing that gets people. This part's a little bit harder. But each unit, every time you go from one number to the next, that's a 10-fold change in the concentration of H plus ions. And I'll give you an example on the next slide. But for now, you need to know that. Every time you move up, it's a 10-fold change. A little bit like the Richter scale, right? It's not like if you go from a 2 to a 1, that's like 1 times worse. It's 10 times worse. And again, I'll give you some examples on the very next slide, but some ones after that. Any questions so far? Here's a picture from the old textbook. It's a good description. Oh yeah, something I didn't say, but hopefully you can guess this, and you probably already know it anyway. If 0 is the most basic most acidic. Let's say that has a pH of 0. And let's say this has a pH of 14, which is the most basic. Then what number would neutral be? Right here, what number is this? If 14 is the most basic you can get, and 0 is the most acidic, then what must neutral be? Good, 7, very good. And you do need to know that too, so sorry I didn't mention that earlier. Yes, 7. And you can see, now we can just kind of ignore those numbers, but you can see this basic solution, right? There's a lot more OH minus ions, right, than there are H plus ions. Here, in the acidic solution, there's a lot more H plus ions than there are OH minus ions. And then here, they're equal. Any questions about that concept? All right, so now let's talk about this tenfold thing, because I'm gonna give you a test question right now, like a version of a test question. So on the test, you might get this right here, so you don't have to memorize this, because I'm gonna ask you a question about it, but I want you to refer to it. It's nothing to memorize. I might ask you something like, how much more, let's see, how much more acidic is grapefruit juice than urine? Right? So then you just have to figure out, and remember that each step is 10. So urine, that's one step to get to 5, another step to get to 4, another step to get to 3. So that's times 10, right? So 5 is 10 times more acidic than 6. 4 is 100 times more acidic than 6. And 3 is 1,000 times more acidic than 6. And of course, if we were to keep going, then it would be 10,000, so on and so forth, right? Each time, you're multiplying it by 10. You guys good on that? Try to remember that. Another thing too, again, like I said, I could ask that question in different ways. Instead of saying, how much more acidic is grapefruit juice than urine? I could say, how much more basic is urine than grapefruit juice? I'm asking the same questions, just using different words. I'll get your question in a second. Another thing I could say is, how much more, how do I put it? How much more of a concentration of H plus ions are there in grapefruit juice versus urine? Or conversely, I could say, how much more OH minus concentration is in urine versus grapefruit juice? And again, every step is a 10 factor. And there's all kinds of different ways that I can ask the same question. So, do you have a question, Carla?
Yes, I was gonna ask, are you gonna ask this to deduct, since we're going up, are you gonna have us, we're gonna have to take away, does that not apply?
I don't know which ones I'm gonna ask, so basically all you need to know is which one's more basic, which one's more acidic, and then just remember every step is a multiple of 10. And to keep it simple, I'm gonna use something straightforward. Like for example, human blood, the pH, even though it's not showing, but you can see it's not quite 7, not quite 8, it's 7.4. Seawater, not quite 8, not quite 9. So I'm not gonna do anything like that. It's gonna be something straightforward, like urine, grapefruit juice, lemon juice, battery acid, right? Something that's right there on there, so there's no ambiguity. I'm not trying to trick you. Just want to make sure that you understand these concepts. And again, we'll do some of this in lab, so you get a little bit familiar with it. Anyway, any questions about this? All right. And again, we are looking at the very simple version, so you can dig deeper for independent work if you want. For example, just this, says battery acid has a pH of about 1, but it all depends, or all these things depend on their concentration. And again, that makes things a lot more complicated, which is why I'm not teaching it, but you can, and it's called molarity, and you can look it up for independent work if you want. But I would recommend only doing that if you're very, very confident that you understand this, because that will definitely muddy the waters. Anyway, if there's no more questions about this, here's a meme that I found online. I think it's funny. Again, I was talking about this last week, or maybe it was Monday. I was talking about dihydrogen monoxide. That's just another word for water. And of course, whoever did this is the group that or page. So this is satire. So it is an exaggeration, but this is the kind of stuff that people will try to fool you with. And in this case, they can try to trick you by using the truth, right? Everything on there is true, but they're just trying to make it seem scary, even though it's not. Dihydrogen monoxide is an acid with a pH level of 7. Well, it's all relative, right? So if anything has a pH level higher than 7, then you could call that this an acid, right? It's all relative. So yeah, it does have a pH level of 7, but as you know, that's neutral, so that's actually not scary. And then to make it even scarier, and again, it's satire, but that's a higher pH level than any other acid. It's technically true, right? Because anything above that would be basic. So if you don't know anything about pH and chemistry, then that could be a scary meme. But if you know just the basics, which is what you're learning now, then you won't be tricked by things like this. Anyway, so the next word for attendance is meme. We're looking at a meme. All right, so we know about acids. We know about bases. Let me back it up a step too. How many of you, well, first of all, like I said here, human blood, the pH is about 7.4. If it gets too much higher than that, and I mean like very little wiggle room, like an 8 I think would be way too much, and 7 would be way too little. If it gets too far away from 7.4, you die, right? You die when your pH, your blood pH goes too far past 7.4. For independent work, you can look that up too. You don't need to know it for the exam. But yeah, how high is too high for your blood, and how low is too low. But anyway, how many of you had coffee or have had coffee? Maybe not today, but in your life, right? Yep, me too. I'm drinking it right now. Swear there's no booze in there. No, yeah. So black coffee, look, I'm drinking it right now, and guess what? I'm not dying. Despite the fact that it's, let's see, from 7 to 10, 100, it's a little bit more than 100 times more acidic than what blood should be, but I'm not dying. How many of you have ever had a soft drink or grapefruit juice, right? And you didn't die, right? Here you are. How about lemon juice? You put some lemon juice in your water, or lime juice in your margarita, right? These things are really acidic. Yet somehow we've had them and haven't died. Which brings us to this concept. And this is what our whole last portion of today's lab is going to be based on. Buffers. You need to know what a buffer is. It's a substance that basically resists changes in pH. So if you have a buffer in a solution, it just makes it harder to change the pH. How does it do that? Well, it just does the opposite of what you do to it. So if you're giving acid to it, it's going to accept those H+ ions. If you're putting a base in there, it's going to donate those hydrogen ions, those H+ ions. So that's how it works basically. A buffer just resists changes in pH. And just so you know, and we're going to talk about it again later in the lab, basically what you're going to do is you're going to have two liquids. One of them is going to be a regular liquid, the other one's going to have a buffer in it. There's also going to be a pH indicator there, so it'll change colors as the pH changes, so that way you can see what pH it is based on the color of it. Anyway, so again, you have those two. Might as well draw it. Yeah, I'll draw it now and leave it, since we'll need this in a couple hours. Two little containers. One of them is going to be buffered. One of them is going to be unbuffered. You're going to drop some acid in there. Actually, I don't remember which one you do first, but either way, you're going to put some acid in and get the pH to go as low as it'll go with some acid, right? Then you're going to put some base in there. See, you can get it as high as it'll go. And you're going to count how many drops it takes to do that. How many drops of acid did it take to get it all the way to the lowest it would go? How many drops of base did it take to get it to go as high as it would go? Hopefully we'll find that the buffered solution takes a lot more drops than the unbuffered solution, right? Because the buffer resists changes in pH, and that's it. That's the basics of what we're going to do at the end of lab today. Now, I've kind of given the TA a little leeway, so I'm not sure what he plans to do. Either you are all going to do this individually, or maybe you're all going to do this in teams, or like I used to do it when it was just me and not a TA. I would just do it, and you guys would watch, and we would do it all together and count the drops together. But anyway, that's what we're doing today. So any questions about buffers? Yeah, we're making good time now. Now we get into, in my opinion, the more interesting portion of this chapter. I'm sure you guys can read that very easily, right? It says biological molecules. So we're done with 2.1, which is the building blocks of molecules. That's what we learned about atoms and protons and electrons and neutrons. Now we just finished 2.2, which was all about water. And in that situation, acids and bases fall into that category of water. And now we're talking about 2.3 biological molecules. In the old textbook, this used to be its own chapter. Again, I'm sorry you can't read this, but here we go. The learning objective. By the end of this, you should be able to describe the ways in which carbon is critical to life. You should be able to explain the impacts of slight changes in amino acids on organisms. For that matter, you should be able to even know what an amino acid is. A lot of people probably don't yet, but you will. you should be able to describe the four major types of biological molecules, and you should be able to understand the functions of the four major types of molecules. Yeah, that's the big one. So these biological molecules, just to go ahead and jump ahead really quick, these biological molecules you're going to learn about, there's four categories of them, and a lot of this portion of the exam is going to be me saying, all right, which of these four biological molecules are we talking about? So I might just give you a specific example, like glucose, what is it? Or I might describe it, say, this thing has a ratio of this many carbons to this many oxygens to this many hydrogens, right? So as we go through this and I'm describing these four different types of biological molecules, remember that's what you need to use in your brain because you're going to get an example on the exam. You should recognize the example and say, oh yeah, that's a carbohydrate or whatever it may be. What I'm not going to do in this 100 level non-biology major course is ask you to just regurgitate all of it, right? So there will be no test question like, tell me everything that makes a carbohydrate a carbohydrate. I'm not going to do that. Again, multiple choice, you just need to recognize things. Anyway, that being said, let's jump into it. Like I already said previously in the chapter, cells are mostly water. Does anybody remember the number that you need to know for the exam? And I don't blame you if you don't remember it, because I only told you once and you probably haven't had a chance to study it yet. Does anybody remember the number that you need to know?
70 through 95%.
Yep, according to the textbook. So therefore, the number that you need to know is 70 to 95%. Make sure you know that. And again, independent work, look that up. What do other sources say? Or for independent work, keep in mind too, that's the average human cell. And the human cells are so different. So if you want to, you can look up specific types of cells and tell me how much water they are. Anyway, the rest of the cell consists mostly of carbon-based molecules. I'm not necessarily going to ask you that. You're probably going to get really sick of carbon by the end of today because you're going to draw a lot of molecules with carbon in them. So all this stuff that you're learning now is like, yeah, you'll definitely know. It's going to be hard to forget that it's all mostly carbon-based because you're going to get sick of them a bit. Why is this a thing? Well, we're going to talk about this bullet point a little bit later. So right now, this is just an introduction. But carbon forms large, complex, and diverse molecules that are necessary for life. So again, nothing to write down, because that's a preview of what we're about to talk about. Organic compounds / macromolecules and carbon-based molecules are the things that we're going to talk about. So really, when you think about it, and you can look this up, I'm not going to ask you on the exam, but again, as far as we're concerned in this course, organic compounds or even organic chemistry, it's all about these carbon-based molecules. So again, for independent work, you can look up what the book says, look up what another source says, and then compare the difference, right? According to the book, this is the definition of an organic molecule. According to this source, this is the definition of an organic molecule. Anyway, this whole slide is nothing but an introduction to what we're about to talk about. So if you're a note taker, nothing to write down there, unless you didn't write down that whole 70 to 95% earlier, because that is the number you need to know. So any questions about this slide? All right. Let's talk about this carbon. Technically, you do need to know this first bullet point, but again, by the end of today's lab, you probably never forget that. But actually, anyway, it's a good preview. So when you're drawing these things, when you're drawing all these molecules in lab today that are carbon-based, that's one of those things you definitely need to know. Carbon makes 4 bonds. So that's how you can check your own work. When you look at your molecule, if every single one of your carbons doesn't have 4 bonds, you need to fix something. Anyway, as far as we're concerned, in our discussion, remember we talked about the big 4, carbon, oxygen, hydrogen, nitrogen. You need to know those, remember the big 4. So that's mostly what we talk about in this portion of the lab and this chapter. But as far as we're concerned, carbon has the most bonds, right? Carbon bonds 4 times. For example, nitrogen only bonds 3 times, which you'll need to know. Oxygen only bonds twice, which you'll need to know. Hydrogen only bonds once, which you'll need to know. So again, this is what makes carbon so versatile, because it has 4 bonds. That's more than the other ones. So because of that, it can make an endless diversity of skeletons. And these skeletons vary in their size and their branching patterns and things like that in their chemistry. And I'll show you an example on the next slide. There are any questions so far about carbon. All right, these are just examples. We'll put a big old X through this. I mean, they're not named anyway, but anyway, you don't need to memorize this. The point here is that we have all these different size and shape molecules. Like, look, this one's pretty straightforward and small, right? This one's a little bit longer than that one, right? And it's a different shape, right? So even though none of you are chemistry majors or chemists, what I can tell you now, what you need to be familiar with, is different shapes, they just do different things. Different shaped things react differently with each other. Because when we think about chemical reactions, what's really happening on the molecular level is these molecules are interacting with each other. They're running into each other, they're bouncing off of each other. In some cases, they're attacking each other. So it's all about the shape. Well, it's a lot about the shape. There's also other things that we'll talk about, but yeah, the shape actually has a lot to do with it. Because molecules do fit in with each other, or repel each other, things like that. So again, these are just a few examples. The point here is that carbon, because of the fact that it can make 4 bonds, it can make all kinds of different shapes, right? Small things, phallic shaped things, a ring. You can see here, sometimes it can do a double bond, right? So this carbon, it's only bonded to 3 other atoms, but it still has its 4 bonds, right? 1, 2, 3, 4. Obviously, all these carbons have 4 bonds, right? This carbon has 4, 4, 4, 4. These carbons, again, this carbon's only bonded to 1, 2, 3 other molecules, but again, it still has 4 bonds. 1, 2, 3, 4. 1, 2, 3, 4, so on and so forth. So any questions about that side? All right. Once again, like I already said a few minutes ago, and then and I think Monday or Friday, it can bond with other elements. Most commonly, so the big 4, carbon is the number 1, obviously, because that's the one we're talking about. But it can most commonly bond with hydrogen, oxygen, and nitrogen. And again, even though this is in the lab, we'll go ahead and give you the hint or the reminder. Hydrogen bonds 1 time, oxygen bonds twice, nitrogen bonds 3 times. You'll need to know that. And of course, carbon, as we know, bonds 4 times. So when you draw these molecules today, that's how you'll check yourself. You're going to look at all the things you drew. Does each carbon, does it have 4 bonds? Does each nitrogen have 3 bonds? Does each oxygen have 2 bonds? Does each hydrogen have 1? Any questions about that? All right, the next word for attendance is gas, G-A-S, because we are talking about methane gas. We put a big old X through this too, because again, this is just an example. I can ask you any questions, but methane is a great example of a very basic carbon molecule, because all it is 1 carbon bonded to 4 hydrogens, that's it. But again, I don't remember if it's your textbook or the old textbook, either way I left it in here. It is very abundant because it's found in natural gas. It's produced by prokaryotes, which are basically, as far as we're concerned, bacteria. Right? So you've got all these bacteria in the swamps. They're decomposing things, and that creates what we call swamp gas, which is methane. It's also found in the digestive tracts of certain animals, right? Animals that eat grass, for example, like cows. As a matter of fact, not that you need to know this, the cows cannot digest grass. They don't have the enzymes. I should either do the green. They don't have the enzymes to break down what's called cellulose, which is something that you're about to learn about, cellulose. But they have bacteria in their gut that can do it, and that's how they can eat grass. Because just like every other mammal, they don't have the ability to break down that cellulose. But in this case, they have stuff in their gut that can. Sort of like termites. Termites, yeah, they eat the wood, but they have things living inside of them that allow them to break down that wood. Oops, excuse me. So any questions about methane before I show you a few more pictures of methane? All right, so there's the structural formula here on the left. You got the ball and stick model in the middle, space filling model. You don't need to know any of that. You will need to be, or not be familiar. The drawings that you're going to do today, this is what you're doing, structural formulas. So almost always in this course, until we get to DNA, then we're going to use mostly structural formulas. So when I try to explain something to you or show you, very often it'll be a structural formula. Actually, I say that, and then on this next slide, it's not, it's the space filling model. But yeah, very often we're going to use structural formulas, not always. So here's an example. Let's talk about some larger organic compounds. So for examples, and these are just examples, so you don't necessarily memorize this. The 2 examples of larger organic compounds. Some of them are the main fuels in gasoline. So right there, that's what octane looks like. Some of them are fuels for your body. So fuel for your vehicle or fuel for your body. And again, if you look at those, remember, like I said, even though you're not a chemist, a lot of times shape has a lot to do with stuff. If something has a very similar shape, it's very possible that it also has a very similar function. And also remember what I said earlier. I think this was Monday or last week. And this is really going to talk about this later. But however much energy it takes to build a molecule, that's how much energy will be released when you break down that molecule. Therefore, when you see something long like this, well, yeah, it probably took a lot of energy to put all that stuff together. If you thought that, you'd be correct. And therefore, if you break it down, there should be a lot of energy to be released, which is why gasoline has so much energy and we use it to move vehicles, and which is why greasy, fat food, has a lot of energy, right? Because those molecules contain a lot of energy. And again, not that I'm going to test you on this, but I want you to really think about this common theme of structure and function. Things that have similar structures very often have similar functions. Of course, as you're also going to learn, sometimes all it takes is a small tweak to the structure to have just a completely different function. And that's something we'll talk about later too. But before I take questions about that, so again, we're comparing a fat to octane. So here's another look at it. That's what octane looks like. And this is what a saturated fat looks like. Of course, the picture I showed you had 3 of those attached to each other, because that's what a fat is, what you'll learn about in this chapter. But yeah, that's part of what makes up a fat that you eat. So you can see how similar they are, right? Again, similar functions. Vehicles break down octane in the presence of oxygen to create energy or to release energy. Your body breaks down fat, among other things, in the presence of oxygen to release energy. So very similar things here. So we're going to now back up a step. So again, to remind you, we're still talking about these biological molecules. So we're talking about all these things that are carbon-based, right? We've got that carbon-based molecule. We have this carbon-based molecule. So again, they're similar, but they're different. It's all about the structure. So the unique properties of all these molecules I'm about to teach you depend on their carbon skeleton and the atoms attached to it. So it's all about the shape of these things and also some of the things that are attached to it. And as you'll see, like so far I've been talking about shape. And that is important. Shape is important. There's also other things to consider, which we're about to talk about. Which brings us to this term, functional groups, which is basically this bullet point right here. The atoms attached to the skeleton, that's the functional group. And as the definition goes from the textbook, a functional group is groups of atoms directly involved in chemical reactions that are attached to the carbon skeleton. So again, very often it's these functional groups that dictate what how these molecules act in chemical reactions. Each one plays a critical role in chemical reactions, and a lot of biological molecules that you're going to learn about have 2 or more functional groups. Here's a list of them. This is not from your textbook, and I will put a big X through this because you don't need to know it. But these are just good examples. So you've got like hydroxyl groups, methyl groups, carbonyl groups, and some of these are going to come back and you're going to see them, but you don't need to memorize any of this. But let's look at it. I mean, look, these all have different shapes, right? This one's kind of straightforward, straight. This one's got some double bonds. That one has some more double bonds. So again, the shape does come back into play. But here's some other stuff that you're going to learn throughout this chapter. Polar and non-polar, they don't mix. So remember, you already know what a polar molecule is, right? That's where the electrons aren't shared evenly. So water, for example, right? The oxygen end is slightly negative. The hydrogen end is slightly positive. Those things don't mix. So in a chemical reaction, if one molecule has this functional group on it, and another molecule has this functional group on it, those things are going to repel each other, like oil and water. Because in that case, oil would be non-polar and water would be polar. That's why they don't mix. Same thing here, right? This one's polar. Another thing that I haven't taught you and you don't need to know, but oxygens are very negative, right? Because again, they attract a lot of electrons. So oxygens are usually very negative. So are double bonds. So this is a very negative thing. Therefore, it's going to be attracted to positive things. And therefore, it's going to be repelled from other negative things, right? So all these things sticking off the carbon skeleton will dictate how it reacts in chemical reactions. Because it'll be attracted to some things and repelled from others. It'll fit with some things in a 3-dimensional way or not fit with some things in a 3-dimensional way. So very much it's all about the shape and the electron situation, right? Is it polar? Is it non-polar? Is it charged? like that one says, right? Which you can't really see it, but anyway, yeah. The carboxyl group is charged. It ionizes to release H plus ions, right? You don't need to know that. The point is it's a full charge. Any questions about this slide, which you do not need to memorize or study? All right, let's make some quick distinctions here, just once, and then after I've made this distinction, then you can brain dump it if you want. But on the molecular scale, a lot of biological molecules are huge, right? And that gives them the name macromolecules. So technically, again, in this portion of the chapter, you're going to learn about 4 categories of molecules, and we will talk about how technically these 3 are macromolecules, and the other one's not technically considered a macromolecule. I will bring it up, I will explain it, I'll mention it, but you don't need to memorize that fact. As far as we're concerned, when we talk about these 4 categories of molecules, as far as we're concerned, you don't need to memorize which ones are macromolecules and which ones technically are not. But yeah, here's the 3 types that are macromolecules. You're going to learn all about carbohydrates, you're going to learn all about proteins, and you're going to learn all about nucleic acids. You don't necessarily need to write that down yet, because we're going to talk about each one of them separately. And again, then there's a 4th category, which are lipids. Lipids, again, are technically not macromolecules, but that's not a fact that I care that you remember for the exam. But yes, carbohydrates, proteins, nucleic acids, and lipids are the things we're going to talk about in this chapter. And you're going to need to know the difference between them for the exam. Before we talk about each of them individually, let's talk about some stuff they have in common. First of all macromolecules are what we call polymers. Polymers are things, or molecules that are made by stringing together smaller molecules, which are called monomers. You need to know that. You need to know what a polymer is. You need to know what a monomer is. And very often in this exam, that's part of what that's what a test question might be. I'll say, if you have a bunch of, amino acid monomers, what is the name of the polymer? Which you probably don't know that yet. But that would be a protein. So if you put a bunch of amino acids together, you get a protein. If you put a bunch of nucleic acids together, if you put a bunch of nucleotides together, you get a polynucleotide. If you put a bunch of, monosaccharides together, you get a polysaccharide. So again, for the exam, this is something you definitely need to think about as I'm teaching this. When we're learning about these macromolecules, remember what the monomers are, remember what the polymers are. That will be definitely on the test. And just so you know, when you're putting these 2 monomers together, we call that a dehydration reaction. So you need to know that too. Dehydration reaction links 2 monomers together. As far as we're concerned, I'll even scratch through that. That's too specific. I mean, that's true, but it's too specific. Just links molecules together. As far as we're concerned in this class, anytime we bring something together in a chemical reaction instead of breaking it apart, that is a dehydration reaction. Why is it called a dehydration reaction? Because it removes a molecule of water, which you don't necessarily need to know, but that might help you, and I'll show you why in the next slide. But yeah, it does remove a molecule of water. That's why it's called a dehydration reaction. Before I move forward, I just really want to make sure you understand this relationship between polymer and monomer. So like a non-biology example, a train, right? If you put a bunch of train cars together, you get a train. So the train cars in that situation would be the monomers. The individual train cars would be the monomers, and the whole train itself would be a polymer. A pearl necklace. Each individual pearl would be the monomer. You put a bunch of them together, that's the polymer, right? The necklace itself is the polymer. That's what we're dealing with. And when we're bringing these together, it's called a dehydration reaction. So here's an example. Here we have a short polymer being added to a monomer, and that's just an example. It could be a monomer and a monomer. It could be a short polymer and another short polymer. It could be a long polymer and a long polymer. It doesn't matter. The point here is, like I said on that last slide, when we bring things together in this class, as far as we're concerned in this class, it's always a dehydration reaction. Let's see. 1, 2, 3, 4, and then 1. So this isn't going to be exactly correct, but let's just say that this chemical reaction is C4, because there's 4 carbons, plus, I don't know. But again, this is incorrect, but I'm just trying to do this quickly to keep things simple for you. C4 plus C gives us 1, 2, 3, 4, 5, C5, right? We'll just say that. But it also gave us water. And that's the important part. This is what I'm getting at. So when you see test question, and you will see it, where I say, is this a dehydration reaction, or there's another one I'm about to teach you. If the water is in the products, like this one, it's on the right-hand side of the chemical equation. If the arrow is pointing to the water, that's a dehydration reaction. Because we took these products, these reactants, and we dehydrated them, and now we have water. So there again, there's a few ways to remember that a dehydration reaction is the one that produces the water. Anyway, any questions about this slide? All right, so again, this is when you bring things together. Any idea, which in my mind, that's actually not that hard to remember. But the next thing I'm going to teach you, unfortunately, might make this a little bit more difficult because of the way that it sounds. But anyway, any guesses to what the opposite of a dehydration reaction might be called? Very good, very close. So if you can remember that, it's actually called hydrolysis. Hydration will make more sense, but whoever came up with it wanted to really make life difficult for biology for students. But no, technically it's called hydrolysis. But yeah, see, I'm glad your brain went directly to hydration, because that does sound like the opposite of dehydration. Basically, what you need to know is that, again, like we just said previously, dehydration reactions break things. So obviously the opposite of breaking things, bring things together. And then hydrolysis reactions break them, right? So they break the bonds between these monomers. They do this by adding a molecule of water, as you can see in this chemical reaction right here. So again, I'm going to incorrectly write this, but what we have is 1, 2, 3, 4, 5, because I'm making this very basic. Let's call that a C5 plus water, right? That's our reactants. And then we get a C3 and a C2. And again, that's really incorrect, but it's okay for what I'm trying to, the point that I'm trying to make here. So when the water is on the left-hand side of the equation, right? When the arrow points away from the water, when the water is with the reactants and not the products, that is called a hydrolysis reaction. We're using water as opposed to producing it. Any questions about that? All right. So again, one more time before we talk about them separately. These are the 4 categories of large biological molecules that you need to learn about. You will need to know their monomers and their polymers. And of course, lipids aren't technically macromolecules, so technically they don't have monomers and polymers, but they are made up of parts, and you will need to know what the parts are called. They don't happen to be monomers. They're not like repeating the same parts. And just for just not out of curiosity, but since we're talking about these large biological molecules, what I'm saying here, what your textbook is saying, is no matter what we know, as far as we know, every living thing has those molecules in it. And I just use those 2 pictures as an example. Does anybody know what this thing on the right is? Not that I expect you to, and not that you'll need to.
Always.
I like that. It kind of looks like it, doesn't it? It's hard to see with this, but what it is the tardigrade. It's one of the smallest animals in the world. You can look them up for, also called a water bear. They're really interesting if you want to look them up for independent work. You can blast them with radiation, put them in a vacuum, freeze them, boil them. Very often they'll survive. They're very tough little things. So that's like, again, one of the smallest animals, which if you want to look that up, another independent work topic. What is the smallest animal?
You said that it's called a water bear.
Yeah, that's one of the nicknames for it, yeah. And then here, this one's a little bit harder to see too, but does anybody know what that is? I mean, obviously it's trees, but does anybody know specifically what type of trees or anything like that? So according to some people, that is the largest organism on Earth. There's a bunch of quaking aspen trees, and they're separate trees, but they're all one organism. So they're all connected at the roots. So if you look at any of their DNA, it's all the same, because that's one organism. And again, another independent work topic if you want to look into it. What is the largest organism on Earth? And again, some sources will give you that answer. Some sources will argue something else. It all depends on how you think about it. Anyway, I just want to use those 2 examples, because again, whether we're talking about the largest organism on Earth or one of the smallest animals on Earth, every living thing has these 4 categories of large biological molecules. Any questions about that? All right. That reminds me. If you download this PowerPoint, I need to make this a little bit more clear. If you click this, you can see that's a little video. It's a 14-minute video. We give you some information about biological molecules. You can watch that if you want. Here we go. Let's talk about the first category, or at least start the conversation. So carbohydrates. When you think of carbohydrates and you're not in biology class, what do you think of?
Bread,
Bread, good. Yeah. What else?
any starch.
Starch, good. Carbs, right? Yeah. Very good. Well, yeah. So it's funny you guys mention that. So there you go. These are great examples, right? So that pasta, right? That pasta, that's a bunch of carbohydrates. I think when most people think of carbohydrates, that's the kind of thing they're thinking of. Like you said, starches, right? But also that Coca-Cola right there. No, I'm not getting paid for that. But that soda with its sugar, a sugar is also a type of carbohydrate. So here's what you need to know. One, sort of. The definition of carbohydrate is they include sugars and polymers of sugars. So even starch, the starch is making up that spaghetti. That's just a bunch of sugars put together. The Coke, Coca-Cola, that actually is the two smaller sugars put together to make another small sugar, but it's obviously a little bit larger. But I'll put a question mark next to this. Is more important than the definition, again, is I'm going to give you an example on the exam, and you're like, yeah, that's a carbohydrate. Of course, to make things a little bit more difficult, as you're going to learn when we come back on Friday, there's different types of carbohydrates. So not only do you need to know the carbohydrates, the monomer is the sugar, and the polymer is the bunch of sugars put together, but you'll also need to know the different types of carbohydrates and recognize them based on their description. Again, like I said, small sugar molecules in soft drinks, that's carbohydrates. The long starch molecules in the spaghetti and the bread, those are also examples of carbohydrates. Let's see. The last word for attendance will be soda, because there's a soda. And then, of course, if you're online, of course, you guys can too for extra credit. You guys can send the words if you want. If you're online, you have to. Either way, do it before 9 a.m. And if you're online, another thing you have to do is include one question, right? So based on all this information I just gave you today, also include one question. And of course, you guys can too if you want those extra credit points. And that's it. I'm gonna do my office hours in here since we have lab in less than an hour. Yeah, that's it. And again, if you can't come to lab this week, don't feel bad about it. You're still just learning how the process works. But if you need help completing either one of those labs that you need to do, let me know. If you came in late, you're probably going to want to email me and tell me you came in late.
I sent my independent work. I know I sent it work.
Probably. Oh, okay. Probably. What I'm doing is, because I'm still behind on grading, kind of prioritizing labs and stuff like that. Independent work. It'll be there. Casebook?
Yes. Also, the words for the other day, I don't know what happened. I sent them after class, like right after you said the word.
Oh, it didn't come in until much later? Well, you can reply to that, tell me. You know, it's fair, it's 0.4 points, so I'm not, but I'm also not too worried about that. Now, if you were absent, Monday, then you have to meet these. All right. So yeah, see, you sent the words, independent work paper. Just got it down, but, oh yeah. So if you have shared it, don't, like via Google Docs, I don't see it yet.
It did.
Yes, what you could do then is just reply to that email.
I think
If you still have it.
I think I just hit share at the top and then I put yours email, so I don't know if that comes up.
I should do it. I should get an email, but apparently I didn't. So if you want to, just send me an email, say, hey, I shared it with you. And then what I can do is go into my Google Drive, go into my shared documents, and once you shared it with me, it'll be there.
Awesome. I will do that. Thank you.
Thank you for following up.